Hi everyone, in this video I'll be talking about how cell wall of bacteria that gives protection against osmotic pressure. Okay, so remember one thing, osmotic pressure means whenever the cell interior means the cytoplasm, okay, the cell interior or the cytoplasm that differs from the environment, okay, in that situation osmotic pressure occurs. Now there are two types of principle here. There are two types of principles, okay. One, that is when a cell, a bacterial cell, okay, that is placed in hypotonic solution. Hypotonic solution means in which the environmental solute, okay, hypotonic solution means environmental solute concentration is less than the cell interior, okay. So hypotonic solution means the environment that contains solutes, but that the concentration of that solute that is much less than the cell interior or the cell cytoplasm of the bacteria which is placed into the hypotonic solution, okay. Now in that case, as we all know that the solute that will move from outside to inside means from hypotonic solution, that is the outside environment, to inside means the cell cytoplasm, okay. Now this will cause a cell to be enlarged because more and more solute will be moved inside the cell cytoplasm, and that cell doesn't have cell wall. Remember that a cell without cell wall, if they are placed in a hypotonic solution, obviously more and more solute that will be moved towards the cell cytoplasm, and that will cause the cell to be enlarged, and finally cell lysis occurs, okay. Now second principle, that is if a cell that is placed in hypertonic solution. Now hypertonic solution means the outside environment has the solute concentration that is much greater than the cell cytoplasm or the cell interior. The cell interior has lower solute concentration. So here also the same principle applies, that the solute that will be excreted or moved from the cell interior to the environment means the hypertonic solution, and that causes the cytoplasm to shrivels or shrinks in such a way that the whole cell from this size it will become to this size, it will be shrinked up, okay. And remember that the cell doesn't contain any cell wall, and that situation is known as plasmolysis, okay. So one is lysis, that is the burst out of the cell due to the enlargement. Why? Because there is more and more solute will be moved inside the cytoplasm, which will make the cell enlarged in such a way that they will burst out because they doesn't have any cell wall. On the contrary, if a cell that is placed in a hypertonic solution, again the solute concentration that is greater in the environment, not inside the cytoplasm. So the bacterial cell that will remove or excrete its all the solutions, all the solutes from the cell interior, that is the cytoplasm, into the hypertonic solution, and that will cause the cell to be shrink from this to this. They will shrink, and that phenomena is known as plasmolysis. So one is lysis, one is plasmolysis. So both of them, that is the cell without the cell wall, whether the gram positive or the gram negative, they always remember that they are sensitive to the osmotic pressure. They either may result in the lysis whenever placed into a hypotonic solution, and if the cell wall, without a cell wall, if a gram positive or a negative bacteria, if they are placed in hypertonic solution, they shrinks, and that phenomena is known as plasmolysis. So this is how the cell wall basically gives protection against the osmotic pressure towards a bacterial cell. On a next video, I'll tell about the lysozymes and the penicillin, that they also have negative impact on the cell wall of the bacteria, whether gram positive or negative, it doesn't matter the type of bacteria, but lysozyme and penicillin, both of them have negative impact on the peptidoglycan layer of the cell wall. And what will be the fate of that cells if they are placed either in hypotonic or hypertonic solution? So keep watching. If you like this video, please subscribe my channel and share with your friends. Have a good day.